Thank you very much, Christoph. It's always nice to participate in this educational program. I will be talking and showing today TMS-EEG experiments with active and passive electrodes, mostly talking about passive, but showing the active. And the reason is that the passive electrodes take a long time to prepare, and Christoph gave me only 1 hour. This presentation involves a lot of work, especially from Alex, who was our intern for a few months, from our colleague Johannes. He was a speaker these days. You probably already saw him, who is our data scientist. Patrick Reitner, who actually designs and is principal developer of the g.tec solution for the Simulink. Our colleague Sebastian, who is equally contributing for the data analysis, and they gave me to present their work here. So, going forward, just very brief introduction about the transmagnetical stimulation. So essentially, we are inducing some current into the coil, as you can see, for instance, on this image, which will produce the magnetic field, which will induce the current in the brain, in the cortex, and that's the main principle. So you could also notice, probably from literature or maybe on the YouTube videos or maybe visiting labs, there are different coils, but you mostly see people using this 8 or butterfly coil for the TMS-EEG experiments, and that is why, because it generates the field which can be better directed at the target point in the cortex. About the EEG, you are hopefully already much more familiar with an EEG after these 3 first days, if not before. Just as a small curiosity, you're looking at the very first EEG device. So all this is the EEG device, and it had only few channels. If you compare that, for instance, to our unicorn, which is probably the size of this knob, you can see how much the technology advanced in 101 years. So another curiosity, this device was actually used for the first time in 1924. However, Hans Bergen made the first communication in Cambridge in 1929, and hence in the literature that is given as the birth of the EEG. So why would we combining the TMS and EEG in the research? That is because we can study very well brain functions and also the connectivity with the propagation of the signal. And as you can see, there are a lot of different specific sub-areas which are mentioned here. You can further investigate it, but in the interest of time, I will keep this part short. As you can imagine, because of the TMS application, it generates a very huge voltage field. Around the electrodes, we would have the electromagnetic artifacts, and then we also have some mechanical artifacts, because sometimes when we use the very high intensity, these can actually cause wires and the electrodes to move slightly. It's not very visible for the human eye, but it does happen occasionally. Then there's a number of the different physiological artifacts, including the eye blinks. Also, experiment takes some time, so there are some head movements, etc. And there is the auditory evoke potentials caused by this click sound or tick sound when the TMS device discharges. So there is also additional artifact that is not presented here, that is recharging artifact, but it is easily actually avoided when you set the recharge to the longer time than your time window. So why are these electromagnetic artifacts so annoying in the EEG. They are annoyed because if you present the whole circuit between the EEG device and electrode here and the brain, we have some structures that actually have certain capacity, and as you can imagine, that would actually prolong those artifacts. So how do we deal with them? So first, about the TMS pulse artifact, we use a specific input box, which is this electrode connector box TMS, which has the limiter circuit in, and which would actually prevent the saturation of the device. And even if we use this kind of the box, we can see over here in this very example that we have still very large pulse artifacts. So this is plus minus 2.5 millivolts. So some channels might have a minor artifact, so to say, although 4 millivolts is not minor artifact, but you could have as much as 50 or more millivolts on the electrodes that are closer to the coil. This is the example recording of one of our clients from Brescia, from Italy. Then we have the possible muscle artifacts, and that is because on the head we have some major muscles which are large in the area, and then could be actually twitched easily with a high intensity coil pass. The way they look like, so they are propagate around 5, or they start around 5 milliseconds. So they have this typical bipolar wave. And this is an example from the field trip manual for the TMS-EEG. Then we have also the auditory EP artifacts, and here is the publication. So you can use these QR codes actually to access the relevant publication or the relevant reference in general. So over here in this article, it's described how do they use the independent component analysis to actually filter out these EP artifacts. And as you can see, they occur between 100 and 200. So we have and 100 and p100 waves that are pretty typical. Then we have our TMS equipment. In our case, we have a bit older model. So there are also different brands, but essentially they all have a coil, a pedal to start the stimulation. We can adjust the intensity and the frequency of the stimulation, and they all have a trigger input out. You would usually have a setup with a chair, which is quite comfortable for the subject. Then we would have the arm, which would enable the better positioning of the coil in terms that you can secure it for a longer time without doing it manually. And we can use a different brand, so we are compatible with any brand that has trigger input and trigger output. Trigger input is interesting for the closed loop circuit, and trigger output obviously to be able to collect TMS-EEG TEPs. And so some of the best known brands are definitely the Next Team, the Mac Venture, Max Team, and so on. So what are the requirements for the EEG hardware when we want to record these experiments? Ideally or obligatory, they should be DC coupled. It means that in the range area, they will actually record it, even if it is not visible. Usually, you would make them easily visible using the filters, but that can be done only for the visualization purposes. And then you can obviously analyze the waves of the recordings data offline. Another thing that is very important is that you have a very high sampling rate. And although 5 kilohertz or higher doesn't seem that high, but if you start multiplying that by 64 electrodes, it's not seldom that you have 1 gigabyte recordings. I have seen some attempts of using the lowest sampling rate, but that was not accepted by the research community. So usually you would be doing 5, 10, or 20 kilohertz, depending on which latency you're aiming for. Then, as mentioned, we need a peak limiter for the input box, and you should be using either passive electrodes, which are preferable for the short latencies, or active electrodes, which can record everything from 10 milliseconds onwards. In our case, we are using our G.tec amplifier, which has the maximum resolution of almost 40 kilohertz, and that can be used on 40 channels, 20 kilohertz, and 64 channels. It's a 24-bit amplifier, so the information is at 22 bits. And for the control, we can use it with a passive, active, and invasive electrodes. There are actually some studies done also with invasive EEG electrodes with TMS. And over here you have the link for our web page for more information. Then we can use different software solutions, like a HiSys Simulink, which is based on the MATLAB Simulink, but we design a lot of these custom-made blocks that allows you to use our amplifiers, also our stimulators, and some of our hardware. That also have a custom blocks that are designed for the manipulation, data processing, like filtering, for the feature extractions like ERT, like FFT, and so on, and many others. So we have nowadays already over 100 blocks, and we deliver the system with more than 100 template models, and this is one of them. This is the model that we are using for the TMS-EEG recording. So this is our amplifier block model. And then we have the signal processing, that is to say, projection of the bed channels. So if you use 64 or 128 channels, it's very likely that few of them, at least one is almost always bad. That happens in every single lab. You can obviously invest a bit more time, and then you can get all of them actually good. Then you can do for instance, the common average reference, apply the notch filter and bandpass filter, and then we have the main block, which is TMS processing blocks, where you define the time window for the analysis. Then we have this post-stimulation interface, or the interval, or the gap, which is essentially the period where of duration of the pulse. So we usually put it very conservatively at 5 milliseconds, and the result is that those 5 milliseconds will be recorded, but those waves will be not painted on the screen. And we can also use the inputs for the E or G channels, so we could detect some artifacts. The another software that we can use is our G-Recorder. It is very user-friendly software, which allows you to see evoke potential window along with the raw data window. And it's easily to follow the development of the experiment, as we will see in our example today. I will be using today the G-Recorder. So the recommended setup is equipment, which in our case is the high ampli with a trigger to our Megan-Moore PowerMac TMS device. Then if we are using the passive electrodes, then we have to keep very low impedance, sampling rate very high. Eventually we can use the common average reference. Then there is a need to actually make everything tidy because of these pulse currents. wires should be as away as possible. So you will see if we have a few wires, we can bund them into one line. But usually with the 64 channels, we have a couple of lines, one for the left hemisphere, the other one for the right. And we should position them perpendicular to coil as possible. Then we should position the coil on the holder arm or the tripod or some other support, and we should preferably use the narrow navigation system so we can position the stimulus as accurate as possible. We unfortunately do not have time for that today. And then we can use the TMS toolbox for simulating that we developed, for instance. In today's example, we can use the G recorder. We will use the G recorder, but as a side note, if we are conducting, for instance, closed loop experiment, then we would need this TMS toolbox. So I will be showing you this on the video. This is how the electrodes should be the wires should be positioned. So we group them by the left and right hemisphere, and their way. So this is example with the passive electrodes, and because of our skin, which is always somewhat greasy, we have actually to clean it, and we clean it usually with abrasive agent. Some examples would be Skin Pure from Koden or NuPrep. And here is a very short illustrative video. How do you do that? So for the passive electrodes, first of all, you should move away the hair laces so you can see the scalp. And you see she's now using the cotton with that abrasive agent, and you firmly clean the scalp. So the sensation is like you would actually pick up a bit sand and rub it against the skin. And I will play this part again. So you see, she cleans it vigorously to lower the impedance. And then when injecting the gel, she will have the tip of the syringe in the air. And why? Because she cleaned only this area. If you press it against the scalp and squeeze it, it's very likely that gel will spread a bit around. And over here, the scalp is not clean, and that would actually ruin your impedance. Also, another curiosity about the impedance, when you prepare the skin, the scalp, usually it takes up to 3 minutes for impedance to settle down. About the coil positioning, it should be at the right spot on the head, and it should be at the optimal distance to the electrodes, correct angle, and heads of the corresponding to the head surface. And by using the narrow navigation, that would be easiest way to achieve it. As you can see, the coil is usually positioned practically against the cap, and that is very important in the case of the active electrode. So these are both examples with the passive electrodes, which have thickness of the 3 millimeters. So how the narrow navigation looks like? So we have usually some markers on the coil, also on the subject. So usually at least 3 points. So you could define the 3D space for the system. Then you have the volumetric MRI from the subject, which is then converted to the realistic 3D model. And then you decide what is your target, which area do you want to stimulate, and you save them. So in this example, we want the target number 3. And while we're positioning the coil, so these are the markers. Then you can with that see on the body, these are the markers of the coil and the coil. And with that, you can see how far from the target is. The newer system also have something that is really like a target. And then the center of the target is a green when you have the short distance, and they're getting red where you are off the target. Another thing about the TEPs and in general about the epoch potentials, you have to be at the right spot to obtain them. So if you are stimulating actually off the site, then you won't get desired result. Likewise, if you are using different intensity, you will have not just the different amplitude of the peak, but also the waveforms can somehow be distorted in the morphology. Not as much, but quite visible, the difference between two. And then also another thing about evoke potentials is, it's very important, is reproducibility. So whenever you record evoke potential, you should obtain at least 2 recordings. You can obtain them at the very same day, in 1 week apart, in 1 month apart, assuming that the conditions are the same. You should get approximately the same results. Another important instrument is the online visualization of the evoke potentials. So this is the system from Professor Marcello Massimini's lab. Silvia Casarotto made this publication in 2022, and a lot of labs, especially in Italy, but also around the world, are using this system, which allows you to see the evoke potentials during the acquisition. So they want to be sure that they are at the right spot. So the X marks the spot where the coin points out, and then you're measuring evoke potential. Then you do at least a dozen of the stimulus to see that you are at the spot, and then you just, if you're happy, continue with acquisition to the large number. We are also developing our online software. It's still in development. It uses the mathematical model that of the electric discharge artifact, and then we subtract it from the data. We will be using in today's example the 64 channel cap. As you can see, we are using the frontal ground and reference. This is very typical layout of the montage, and we are also covering all areas of the brain, because if you are studying the propagation, then you want to compare signal or AP at the different points. Now, we have been developing, you probably see, who followed us the last year, you have noticed that we were using some red electrodes, which were a bit thicker, and we were developing these active electrodes, which are now 3 millimeter sticks, so just as the passive electrodes. And you can see that we can actually come very close to the head, just like with passive electrodes. Now, the small difference between these electrodes and passive electrodes is that they have some electronic circuit in, which kind of pre-amplify the signal, and that would actually speed up the preparation time, which is important. So we shall see today in 10 minutes. I'm supposed to prepare Christoph, who will be my subject today, for the recording. And on the opposite side, because it does have this electrical circuit inside, it actually has the larger pulse artifact. So with our passive electrodes, we get the pulse artifact around 1.5 milliseconds. It's very short. And with these electrodes, we get 7 to 10 milliseconds delay artifact. Now we shall proceed with a live demonstration. And this is done in the February. So you see that Christoph is willing subject over the time, helping us develop the solution. Let's hope he won't be jumping today. And this is the recording from the last month. As you can see, we were using our online TEPs in this case. And with that, we shall pass now to the live recording. Okay. I will also switch to another camera. Just a moment, please. So hopefully you can see me now. So we have a setup here with a TMS device. So over here we set up to 60% of the intensity. We can adjust for higher or lower intensity. We can also adjust the frequency of the pulse. Then we have the trigger output that is connected to our amplifier. So this is this green cable coming in. That's how we will trigger. And then we have our box and the cap there. So we will not be using this chair, which has the wheels. That is something normal. We want for our subject to be, please, Christoph, to be comfortable. And maybe I can ask my colleague to film me while I'm walking around to show you what does it take. So thank you, Bastian. The first thing we can do with these electrodes is to put a bit of the alcohol on the cotton pad and just degrease the scalp in general. You don't have to do that. We made successful recordings even without it, a bit more alcohol. Whenever you're using something potable like alcohol, start rubbing with the central or posterior area so it wouldn't go to the eyes eventually. So you do vigorously, and Christoph has more hair. Okay, that should be enough. Okay, two bits. Then the scales to have predetermined positions. You are wondering, sorry, you are worrying about two things. First is the position of the electrode, which is this one, number 30. So it should be in between the ears and on the crossing of the imaginary line between the nasia and the inion. Inion is this bone at the back. So if it is at the position, you're happy with it, and we will strap the subject. You can also measure it, but as you have seen, Christoph is frequently the subject, so I have the target in my memory. Now, and we will open the G recorder. Takes a minute. And the gel. Now we are going to open the impedance measurement tool. Okay, so yeah. So we made the custom montage for it, and now we can use it in the impedance measurement. Okay, so we have to tell the system that we are measuring with a TMS. We can make this whole bigger and show the labels clearly. And for the measurement of the impedance, we have to prepare first the ground.
Sebastian, can you film again?
The ground and the first electrode, which is the electrode used for impedance measurement. And then we go likewise to all other electrodes. As you can see, this is going fast. Measurement. You can go whatever way you like. I like to go by order. So 4, 5 horizontally, but you can also do the preparation longitudinally. This does not have air here, but it's easy. So it does take some time to measurement.
The
Reference is here. Yes. Because this is the part 5 for the info box. Then when it's working, it has the LED lit on. Now we already see the impedance for the electrode we already tell. As you can see, this goes very fast.
I also see everything is black, like the Hans Berger original EEG machine.
Exactly.
We decided to follow the Hans Berger scheme, like everything is black.
Now, as we are preparing electrode by electrode, you can say that the measurement is appearing with some delay, which is normal. So it takes few seconds from the moment that you inject the gel up to the moment that you see the measurement. Now over here, there is already some air, and then you try to move it away from the electrode like this. You're making this linear movement. And you can see the FP7 and FP5 already have very good impedance. Today also very collaborative subject. So around these 5 minutes, we already prepared almost half of the electrodes. No, there is not. So as you see over here, we bundled all the wires to the single bundle, just like you have seen in the picture that I have shown for the passive electrodes, so that part is safe. We don't want wires to be affected by the TMS pulse.
you try and speak louder?
Oh,
They're
okay.
saying it's a little bit quiet.
A bit of cell running away. So again, we are now at the area where Christoph has more laces, hence I'm doing these movements. Over here, this electrode floats a bit, number 26. So in those cases, you can use the tape, or you simply inject a lot of gel, like I did. So T7 is now green. When you're injecting more gel, you should be very careful how much more. It's not very difficult. You need just some practice. And as I'm doing this for some time now, I can do that quite quickly, and I know what to expect, but you can achieve the same thing in no time, practically. So again, we are moving away a bit here, and then we inject the gel. So you see this electrode is also floating away. You can see by the movement of the cap here. You can see that the electrode is over here. So I will inject again a bit more gel, and I'm looking for it to cover the electrode like that. And with that, we almost have everything ready. Even now, I don't know how to adjust it on the mobile. Sorry, you can maybe.
I could try and switch this one to the laptop microphone.
It's almost done. So we are now doing the posterior line, 2078. Yes, thank you. So now we're doing posterior parieto-occipital electrodes, PO8, PO4, this is POZ. I'm doing at the moment. And as you can see, the measurement is slightly delayed, but it's over there. So this was PO3, PO7, 1. So left occipital, center occipital, OZ, and PO2, which is the right occipital. Again, moving away here. Now we're looking at a few electrodes that are not so good. So this C2. I'm slightly scrubbing with a syringe tip Christoph scalp. And as you can see, it's going down. Now this central line is very important, so you would be actually trying to get it as low as possible. And also this FCZ. And with that, we have it basically like this. Okay, so now everything is ready. The next step would be the point positioning. So here we have our arm. We are looking at the motor cortex on the left side. Another thing that is important, you will notice that we have everything on the left side that is in the TMS area. Everything that is on the left side is the TMS, and everything that is G is on the right-hand side. Maybe I can show that. So you see the TMS device, sorry, EEG amplifier is over here, the input box, while everything else is on the other side. And now perhaps we can video here. And let's position the coil. And I can use maybe, sorry, the video from the other one. So let's see. Exactly. And now I will try to position the coil just above the motor area. As you can see, it's still a bit high. And maybe like that. So we are looking for the 45 degrees angle. TMS is getting ready. Okay, so we have the movement. Once we have a movement, we can actually ask the subject, in this case, Christoph, to pose it down. So thank you, Christoph. Subject should be relaxed, seated comfortably, without any muscle contraction. That is valid for legs, arms, but since Christoph is very experienced, there is no need for special instruction. You would need to do a normal subject, usual subject. Okay, so if you blink, yes, because we have the frontal reference, we have blinks everywhere. If you close your eyes, you're looking for some alpha on the back. There was something there, yes, thank you. And if you clench your teeth, we should see the empty artifact or the EEG, okay? And we can start the acquisition. So you will see over here, this online artifact is pretty big, but what is important is to count the trials. So we have already 3 trials, 4, 5. And so on. So how are we doing with the time? I'm almost done. So usually you would make like 100 of these trials or more. People do usually between 1 and 200, depending on the area they're recording and the signal quality. But this is essentially how you do the CEP acquisition. Okay, so we already have over or around 40 trials. That means that in less than 20 minutes, we actually perform the experiment. As a comparison, 10-10 preparation of 64 channels with a passive electrode, just the preparation would take 40 minutes for experienced researcher. Okay, so we won't bother Christoph much more. We will take off the coil. And I will pass to another screen. So hopefully you can hear me again. I will stop the acquisition and close the program. Because if it didn't stop. Okay. So continuing our presentation, in the offline analysis, we would need to remove the EOG artifacts. You have noticed that whenever Christopher was blinking, there was a large artifact in all of channels. Then you should reject bad channels. That happens, as I mentioned. However, when you have the target area, like in our case, the motor area, so you have noticed that I was having initially the high impedance on the C2 electrode, you should actually prepare that electrode the best as possible. if necessary, even to change it, because that's the very important in that case. And then we would be using the epoching. We noticed that we have recorded the TMS trigger, and that would be our zero point of time for the evoked potentials. Then we would do some interpolation of the missing data. Usually we can manually reject trials, the trials that are very noisy. You can inspect it visually and erase them. You can use the ICA for the muscle and other artifacts removal, for instance, for the auditory, as we have seen, and filter, re-reference, and correct the baseline. And then you will get the results. That is also time-consuming process. And when you perform everything, this is the previous recording. And as you can see, we have a very nice evoked potential obtained from the subject. And this is just a note that we have done the extensive testing of our active electrodes. So we were using some passive electrodes and some active electrodes. So active electrodes are these in reddish color, and the passive one, which are so far gold standard, in the green one. And as you can notice, from the measurement, the result is practically the same. So there will be soon an article published about this, and it's essentially the confirmation, the validation of the technology that we are now employing. We would like also to thank the previous interns, Antonio Calzone and Giovanni Lombardi from University Federico II in Napoli, and special thanks to Marta Bortoletta, who was before in Brescia, now is in Firenze. And with that, I conclude the presentation, and I'm open for eventual questions.
Slobby Basileus is asking, can you do such experiments with dry electrodes, or what's the problem?
So dry electrodes have a very high impedance. They cannot be measured, essentially. And because they have a high impedance, they are very susceptible to the ambient artifacts. And as you can imagine, even more to the pulse artifact from the TMS. In short, the answer is no.
And Chandra wants to know, does each active electrode have a preamplifier?
Yes, every single one of them. The only passive electrode on that cap is the ground.
And why does it take about 3 minutes for the EEG to stabilize?
The measurement of the impedance. So it's probably the chemical process when you have this gel introduced on the scalp of the subject. That is something that we observe, but we didn't measure the behind process. So I would explain it like that. When you introduce to different materials that start merging with each other, it takes some time to level down to get the balance.
Slobby, the questions are getting more and more, so I would like to ask you to answer them in the Q&A so that we can move to the next speaker. Thank you very much for the nice demo.